Continuation of the previous video about immunity. So antibody dependent cell mediated cytotoxicity or ADCC. The NK function is to destroy malignant cells and cells infected with microorganisms. They recognize their targets with the help of binding with antibodies. They can bind to antibodies that coat infected or malignant cells. Thus, the antibody bridges the two cell types. This process is called antibody dependent cell mediated cytotoxicity and can result in the death of the target cell. Next topic, B cell maturation. B lymphocyte precursors, progenitor B cells or pro B cells develop in the fetal liver during embryonic life in the bone marrow, afterwards continuously throughout life. Pro B cells develop into precursor B cells or pre B cells which synthesize cytoplasmic IgM. The mu chain and surrogate light chains of the pre B cell are expressed at the cell surface with two closely associated transmembrane molecules known as Ig alpha and Ig beta, which are disulfide linked to each other. At this point, the cell is called an immature B cell. After leaving the bone marrow, the cell starts to produce cell surface IgD molecules as well with the same antigen binding site as the IgM molecule. It is now called a mature B cell. So, it is this cell that can respond to foreign antigens in peripheral lymphoid organs. Here you could see a schematic representation. The main stages of B cell development: stem cell, pro B cell, Ig alpha, Ig beta, mu chain, surrogate L chain, pre B cell, then immature B cell, mu chain, L chain, IgM, IgD, alpha chain, mature B cell. B cell receptor, Ig alpha and Ig beta are associated with membrane Ig molecules on all cells in the B lineage, from the pre-B cell to the memory B cell. The complex of Ig alpha and Ig beta associated with membrane Ig molecules of more mature cells in the B lymphocyte lineage is known as B cell receptor. Ig alpha and Ig beta do not bind antigen. Their function is to transmit signals to the cell nucleus, leading to a change in the pattern of genes expressed. For this reason, Ig alpha and Ig beta are referred to as signal transduction molecules. Pre-B cell receptor, other molecules on the B cell membrane like CD19, CD81, also known as TAPA1, and CD21, CR2 are associated in a complex known as B cell co-receptor, also affect the signal that is transmitted through the BCR. B cell co-receptor. CD, cluster of differentiation molecules, are cell surface proteins present on lymphocytes, both B and T cells and other cells of immune system. To date, over 300 CDs have been characterized. CDs have both biological and diagnostic significance. The presence of various CDs on cell surface can be used to determine the cell's identity. B cell also express surface proteins known as MHC class II molecules. Unlike the expression pattern of many other cell types, B cell expression of MHC class II molecule is constitutive, that is, the molecules are always expressed. Activated B cells also express high levels of a family of molecules known as B7 and are always expressed. Then CD18 to CD86, which are referred to as co-stimulatory molecules because they are required in addition to antigen to activate naive T cells. Resting mature B cells express low levels of B7 and are poor APCs, whereas activated B cells are very efficient APCs. In contrast with its appropriate antigen, the mature B cell undergoes clonal proliferation. Some activated B cells become long-lived memory cells. The majority of activated B cells are transferred into plasma cells. Plasma cell is the antibodies secreting cell which continuously secrete antibodies at the astonishing rate of about 200 molecules per second. A plasma cell makes an antibody of a single epitope specificity of a single Ig class and allotype and of a single light chain type only. And B cell activation require antigen. Depending on the nature of an antigen, B cell activation proceeds by two different pathways: Th cell dependent and Th cells independent. In Th cell dependent pathway, Th cell and B cell cooperate in presence of thymus dependent antigens. Thus, the B cell response to thymus-dependent antigen require direct contact with Th cells, not simply exposure to Th-derived cytokines. In Th cells independent pathway, antigens called thymus-independent antigens activate B cells without direct participation of Th cells. Thymus-dependent antigens are generally large polymeric molecules with multiple repeating antigenic determinants. For example, the components of bacterial cell walls such as lipopolysaccharides and the capsule polysaccharide components. Polysaccharide components associated with the MHC molecules. The double positive cell likely interacts with MHC and peptide expressed on the dendritic cell surface. A T cell expressing a TCR that reacts with too high an affinity to the combination of MHC and peptide is deleted by apoptosis. Thus, negative selection removes T cells expressing TCR with high reactivity to self components that result in self tolerance. So, precursor cell diversification of T cell receptors. No TCRs expressed do not interact with MHC, death by apoptosis. TCRs with no recognition of self MHC and self peptide, death by apoptosis. Then TCRs with strong recognition of self MHC and self peptide, negative selection, signaled death by apoptosis. Then TCRs with weak recognition of self MHC, self peptide, positive selection, signaled survival, maturation to peripheral lymphoid organs. Positive and negative selection in the thymus. Thymic selection involves thymic stromal cells, epithelial cells, dendritic cells, and macrophages, and results in mature T cells that are both self-MHC restricted and self-tolerant. Adapted from molecular biology, this picture. Okay. So, current evidence indicate that peptides expressed by cortical epithelial cells play a critical role in the positive selection step. These peptides are derived from self-antigens either expressed in the thymus or brought in the thymus. It is not currently clear, however, how these peptides derived from self-antigens select T cells with TCR specifies for non-self as well as self-antigens. In addition, it is not clear whether the peptides expressed by the cortical epithelial cells in positive selection differ from those expressed by the dendritic cell in negative selection. Then double positive cells that survive negative selection downregulate expression of either TC4 or TC8 by a mechanism that is currently not well understood. This results in the development of either CD4 plus or CD8 minus or CD4 minus CD8 plus single positive T cells. And function of T cells. If a new T cell that has not yet encountered antigen recognizes an antigen MHC complex on an APC or target cell, it will be activated, initiating a primary response. APC provide two kinds of signals for activation. Signal 1 is provided by a foreign peptide bound to an MHC protein on the surface of the presenting cell. This peptide MHC complex signals to the T cell receptor and its associated proteins. Signal 2 is provided by co-stimulatory proteins, especially the B7 proteins, which are recognized by the co-receptor protein CD28 on the surface of T cell. The expression of B7 proteins on an antigen presenting cell is induced by pathogens during the innate response to an infection. The new T cell enlarges into a blast cell and undergoes repeated rounds of cell division. It generates a large clone of progeny cells which differentiate into memory or effector T cell populations. The effector T cell carry out specialized functions such as cytokine secretion, B cell activation, and cytotoxic killing activity. Then function of T helper cells. Effector T helper cells can be divided into two subsets based on the different cytokines they produce. So Th1 cells synthesize and secrete IL-2, IFN alpha, and TNF beta, which are responsible for delayed type hypersensitivity and the activation of cytotoxic T lymphocytes. NK cells and macrophages. Th2 cells synthesize IL-4, IL-5, IL-10, and IL-13, which functions more effectively as a helper for B cell activation. And function of T cytotoxic cells. The principal function of T cytotoxic cells is to kill cells that have been infected by bacteria and viruses. T cytotoxic cells are also involved in killing transplanted foreign cells during graft rejection and tumor cells. For this reason, T cytotoxic cells are frequently referred to as killer T cells or cytotoxic T lymphocytes. The cell killed by a CTL is known as a target, which can be specialized antigen presenting cells such as a dendritic cell or by other cell in the body. The TCR of a CTL recognizes a combination of peptide and association with MHC class 1 molecule on the surface of a cell. This interaction in the presence of appropriate second signals results in the death of the cell presenting the peptide. CTL also synthesizes cytokine like IFN gamma, which regulates certain viral and bacterial infections, as well as INF beta, which plays a role in target cell killing. Then CTL killing of target cells. Once activated, mature CTL initiates killing by attaching to the target cell. Killing by CTL is thought to occur by two pathways. The first predominant pathway involves the activation of the cytotoxic substance contained in granules inside the T cell. After attaching to the target cell, the CTL mobilizes its granules directionally toward the target and releases the contents of these granules onto the target cell. The major constituents of the granules involved in target cell killing are perforin and granzymes. Perforin is a molecule that polymerizes to form transmembrane channels or pores in the target cell membrane. The action of perforin on cell membranes is similar to the complement membrane attack complex described later. So CTL killing via this pathway also involves granzymes, a set of serine proteases. Granzymes pass on to the target cell through the pores and interact with intracellular components of the target cell to induce apoptosis. Here you could see perforin entering target cell, perforin channel, granzyme triggers apoptosis of target cells. The second pathway of target cell killing occurs via the interaction of the molecule Fas-ligand on the CTL with Fas, a surface molecule expressed on many host cells. This interaction activates apoptosis of the target cell. The cytosolic tail of the Fas contains a death domain which, when activated, binds to an adapter protein which in turn recruits a specific procaspase or procaspase 8. So clustered procaspase 8 molecule then cleave one another to produce active caspase 8 molecule that initiate the proteolytic caspase cascade leading to apoptosis. Here you could see that Fas activated procaspase 8 adapter molecule target cell, activated caspase 8 caspase cascade apoptosis of target cells. So summary of properties of T cell: origin bone marrow, maturation thymus, expression of AG receptor thymus, differentiation lymphoid tissue, surface IG absent, immunity cell mediated as well as humoral, second secretory product cytokines, receptor TCR on membrane. The next, the complement system. The complement, term coined by Paul Ehrlich system, made of approximately 30 circulating and membrane expressed proteins, is an important effector arm of both the innate and antibody mediated acquired immune response. The most important function of the complement system are lysis of cells, opsonization, inflammatory responses, and immune clearance, which removes the immune complexes from the circulation and deposit them in spleen and liver. Complement components are designated by numerals C1 to C9 by letter symbols factor D or by trivial names homologous restriction factor. So designation C1, C2 up to C9 denotes the order in which the components were discovered rather than their position in the activation sequence. So that is just there. The order in which the components were discovered. So complement components are synthesized mainly by liver hepatocytes. In the circulation, the concentration of all complement proteins is about 3 mg per ml. Some complement components are found at high concentrations, while others, such as factor D and C2, are found only in very trace elements or amounts, trace amounts. Trace amounts. Then complement action pathways. Most complement components present this serum in functionally inactive forms as proenzymes or zymogens. Complement activation pathways involves the activation of one component that triggers the activation of the next component in the sequence. Upon activation, individual components are split into fragments designated by lowercase letters. The smaller of the cleaved fragments is generally designated with a lowercase and the larger fragment with a lowercase b. For historical reasons, however, the larger cleaved fragment of C2 is usually referred to as C2a and the smaller fragment C2b. Okay. The complement fragment interact with one another to form functional complexes. Those complexes that have enzymatic activity are designated by a bar over the number of symbols. For the three pathways of complement activation, there are classical, lectin, and alternative pathways. So the classical pathway was so named because it was the first complement pathway to be worked out. The classical pathway commonly begins with the formation of antigen-antibody complexes. Thus, this pathway is a major effector pathway of the humoral adaptive immune response. Other activators include some viruses, necrotic cells, and subcellular membranes. From mitochondria, aggregated immunoglobulins and beta amyloid found in Alzheimer's disease plaques. Other activators include some viruses, necrotic cells, and subcellular membranes from mitochondria, aggregated immunoglobulins, and beta amyloid found in Alzheimer's disease plaques. The classical pathway is initiated when C1 binds to antibody in an antigen-antibody complex. C1 is the complex of three different proteins, C1q, made of 6 identical subunits combined with 2 molecules each of C1r and C1s. IgM and certain subclasses of IgG, human IgG1, IgG2, and IgG3 are efficient complement activating antibodies. Among the human immunoglobulins, the ability to bind and activate C1 is in decreasing order like this. Just look at this figure. IgM greater than IgG3, greater than IgG1, very greater than IgG2. So IgG4, IgD and IgA and IgE do not have C1q receptors. These antibodies do not bind or activate C1 and thus do not activate the classical complement pathway. Then the lectin pathway. Lectins are proteins that recognize and bind to specific carbohydrate target. The lectin pathway is activated by terminal mannose residues of glycoprotein or polysaccharides found on the surface of bacteria. This pathway does not depend on AB for activation. Lectins are proteins that recognize and bind to specific carbohydrate target. The lectin pathway is activated by terminal mannose residues of glycoprotein or polysaccharide found on the surface of bacteria. This pathway does not depend on AB for activation. It is activated by binding of mannose binding lectin and acute phase protein to mannose residues. These terminal mannose residues are not found on the surface of mammalian cells, and so the lectin pathway of complement activation may be responsible for discrimination between self and non-self. Because this pathway is activated in the absence of antibody, it is part of the innate immune defense. Then the alternative or properdin pathway. The alternative pathway is antibody independent. This pathway of complement activation is triggered by almost any foreign substances. The most widely studied include lipopolysaccharide from the cell walls of gram-negative bacteria, the cell walls of some yeasts, and a protein present in cobra venom known as cobra venom factor. Some agents that activate the classical pathway, viruses, aggregated immunoglobulins, and necrotic cells, also trigger the alternative pathway. Activation of the alternative pathway occurs in the absence of specific antibody. Thus, the alternative pathway of complement activation is an effector arm of the innate immune defense. This major pathway of complement activation involves 4 serum proteins, C3, factor B, factor D, and properdin, also known as factor P. So, classical pathway AB dependent, AGAB C1 binds, C4 binds, C2 binds, C4A, C2B, formation of C3 convertase. Then lectin pathway, alternate pathway we could see. And activation of C3 and C5. C3 cleavage is a critical step in all three complement pathways. The C3 convertases of the classical and alternative pathway cleaves C3 into two fragments. The smaller C3A is a fluid phase anaphylatoxin. The large fragment C3B continues the complement activation cascade by binding to the cell surface around the site of complement activation. So anaphylatoxin bind to receptors on mast cells and blood basophils and induce degranulation with the release of histamine and other active mediators. Anaphylatoxin also induce smooth muscle contraction and increased vascular permeability. C3b binding to either the classical or alternative pathway. C3 convertases allows the next component in the sequence, C5, to bind to be cleaved. For this reason, the C3 convertases with bound C3b are referred to as C5 convertases. The cleavage of C3 produces two fragments. C5a is released into the fluid phase and is potent anaphylatoxin. C5b binds to the cell surface and forms the nucleus for this binding of the terminal complement components. And the terminal pathway, the terminal components of the complement cascades are C5b, C6, C7, C8 and C9. These components bind to each other sequentially to form a macromolecular sequence called membrane attack complex. This results in cell lysis. The three complement pathways converge at the membrane attack complex. This complex forms a large channel through the membrane of the target cell, enabling ions and small molecules to diffuse freely across the membrane. The first step in MAC formation is C6 binding to C5b on a cell surface. C7 then binds to C5b and C6 and inserts into the outer membrane of the cell. In the terminal pathway, the terminal components of complement cascades are C5b, C6, C7, C8 and C9. Those components bind to each other sequentially form a macromolecular structure called the membrane attack complex or MAC that results in cell lysis. The three complement pathways converge at the membrane attack complex. This complex forms a large channel through the membrane of the target cell, enabling ions and small molecules to diffuse freely across the membrane. The first step in MAC formation is C6 binding to C5b on a cell surface. C7 then binds to C5b and C6 and inserts into the outer membrane of the cell. The subsequent binding of C8 to C5b67 results in the complex penetrating deeper into the cell's membrane. The final step in formation of the MAC is the binding and polymerization of C9, a perforin-like molecule, to the C5b678 complex. This complex on the cell membrane acts as a receptor for C9 that binds to C8. Additional C9 molecule react with the C9 molecule in the complex to form polymerized C9. This poly C9 forms a transmembrane channel that disturbs the cell's osmotic equilibrium. Ions pass through the channel and water enter the cell. The cell swells and the membranes become permeable to macromolecules which then escape from the cell. The result is cell lysis. Then explain why serum IgM cannot activate complement by itself. Serum IgM is in a planar form in which the complement binding sites in the Fc region are not accessible. Only after binding to antigen does IgM assume a concentration in which the complement binding sites are accessible. And hypersensitivity. Hypersensitivity is an exaggerated immune response that results in tissue damage and is manifested in an individual on a second or subsequent contact with an antigen. So hypersensitivity has been traditionally classified into immediate and delayed types based on the time required for sensitized host to develop clinical reactions on re-exposure to the antigen. Later, Gell and Coombs proposed a classification scheme which defined 4 types of hypersensitivity reactions. Type 1 hypersensitivity. Type 1 hypersensitivity, also known as allergic reactions. Reactions are rapid, occurring within minutes after the re-exposure to antigen. Thus, allergic reactions are also called immediate hypersensitivity. These IgE-mediated reactions are stimulated by the binding of IgE, its Fc region, to high affinity IgE, specific Fc receptors expressed on mast cells and basophils. When cross-linked by antigens, the IgE antibodies trigger the mast cells and basophils to release primary inflammatory mediators stored in the granules. These modulators cause all the normal sequences of an acute inflammatory reaction: increased vascular permeability, smooth muscle contraction, granulocyte chemotaxis, and extravasation, etc. So, mast cell activation IFC also leads to the production of two other types of mediators. These secondary mediators, unlike the stored granule contents, must be synthesized de novo and comprise arachidonic and derivatives, prostaglandins and leukotrienes, and proteins, cytokines, and enzymes. So, mast cell binding fragment, Fc receptor for IgE, subsequent exposure to antigen sensitized mast cell, then release of allergic mediators. The next, type 2 hypersensitivity. Type 2 hypersensitivity is generally called a cytolytic or cytotoxic reaction because it results in the destruction of host cells either by lysis or toxic mediators. Type 2 hypersensitivity is caused by antibodies binding to cells or tissue antigens. The antibodies are of IgM or IgG classes and cause cell destruction by Fc dependent mechanisms, either directly or by recruiting complement via the classical pathway. So, classical example of type 2 hypersensitivity reactions are the response exhibited by a person who receives a transfusion with blood from a donor with a different blood group and erythroblastosis fetalis. This is type 2 hypersensitivity. Two different antibody mediated mechanisms are involved in the cytotoxic reactions. In complement mediated hypersensitivity reactions, antibodies react with a cell membrane component, leading to complement fixation. This activates the complement cascade and leads either to lysis of the cell or opsonization. Blood cells are most commonly affected by this mechanism. So, complement activation is also there. An antibody dependent cell mediated cytotoxicity, ADCC, used Fc receptors expressed on many cell types, natural killer cells, macrophages, neutrophils, eosinophils, as a means of bringing these cells into contact with antibody coated target cells. Lysis of these target cells require contact but does not involve phagocytosis or complement fixation. Instead, ADCC lysis of target cells is analogous to that of cytotoxic T cells and involves the release of cytoplasmic granules containing perforin and granzymes. Their activate events leading to apoptosis. So, ADCC reactions involve IgG and IgE Fc receptors. Then type 3 hypersensitivity. Type 3 hypersensitivity is mediated by immune complex essentially for IgG antibodies with soluble antigens. Circulating immune complexes may lodge at various tissue sites where they activate complement and subsequently cause tissue cell lysis. Normally, these complexes are phagocytosed effectively by the fixed monocyte and macrophages of the monocyte macrophage system. In the presence of excess amounts of some soluble antigens, the antigen antibody complexes may not be efficiently removed. Their accumulation can lead to hypersensitivity reaction from the complement that triggers a variety of inflammatory process. This form of hypersensitivity has a lot in common with the type 1, except that the antibody involved in IgG and therefore not performed to mast cells. Then type 4 hypersensitivity. The type 4 hypersensitivity, commonly called delayed type hypersensitivity, is the only class of hypersensitivity sensitive reactions to be triggered by antigen specific T cell mediated immunity reactions. This is mediated by T cell dependent effector mechanisms involving both Th1 cells and cytotoxic T cells. So, antibodies do not play a role in type 4 hypersensitivity reactions. On activation, the Th1 cells release cytokines that cause accumulation and activation of macrophages, which in turn cause local damage. And autoimmunity, which we will be learning tomorrow, but just have a quick intro. The body is normally able to distinguish its own self antigens from foreign non-self antigens and does not mount on immunologic attack against itself. This phenomenon is called immune tolerance. Autoimmunity is a condition in which structural or functional damage is produced by the action of immunologically competent cells or AB against self antigen. Autoimmunity literally means protection against self, but actually it implies injury to self, and therefore sometimes the term is also under criticism. So, autoimmune disease result from the activation of self-reactive T and B cells that, following stimulation by genetic or environmental triggers, cause actual tissue damage. Four factors influence the development of autoimmune disease. These factors are genetic, viral, hormonal, and physico-neuroimmunological. The influence of stress and neurochemicals. All four of these factors can affect gene expression, which directly or indirectly interfere with important immunoregulatory actions. So that's all about this. Tomorrow we are going to learn about autoimmunity in detail. AI, I guess. AI and immunology, not artificial intelligence, autoimmunity. That's sort of short form. So thank you for listening.